Okay, so here we have a concept about potential. And what we have here, again, creating this potential for what? Potential to do work. We've had this before, right? We've had this in a mitochondria. I'll make a another review thing, right? When we created a proton gradient, lots of protons in the intermembrane space, not as many in the matrix, right? This gradient caused this system to have the potential to do work. What kind of work? When these protons diffused into the gradient, into the matrix along their gradient, they went through these little lollipop structures. Now my drawing's not great. That did the work of phosphorylating ADP to make ATP. That's one place where we saw a gradient, and this gradient also was of charged particles, like we're going to see, but it's not going to be protons here, right? We have a potential to do work that has to do with charges, all right? And here is the basic idea for these nerve cells. Here's a standard nerve cell body. There, I'll put an axon in there and some dendrites, right? What it says here are cations, positive ions, are more concentrated outside than inside. Therefore, relative to the outside, the inside is negative. It doesn't mean it's overall negative. It just means it's more negative than the outside relative to. This is the potential we're talking about. And these charges are attached to ions, or they are charges that are actually ions. And in this case, it's going to be sodiums and potassiums, and then to a more minor degree, chlorines that we're going to talk about, but mostly sodiums and potassiums. Now you say, wait a minute, those are both positive. And that's true. But what we're saying here is that outside of a neuron, there are more positives than there are inside the neuron. Therefore, there's more negatives inside than there are outside. Okay, so this potential to do work, when this thing is at rest, this is again a neuron, a very simple one, dendrites, axon, right? This potential to do work is called its resting potential when it's not doing work, right? So here we have that term resting potential, and that's what I just described. What's the resting potential? It is that the inside is more negative compared to the outside, okay? How does that get maintained? Because we have a gradient of charged particles, how come they don't diffuse in and out and destroy that gradient? This is the sodium-potassium pump doing this. It's keeping this gradient of more positive outside than inside, okay? Well, how now does this cause this nerve cell to be able to do that action of sending a message? Well, this action of sending a message that's due to this potential difference is called an action potential. And what's going to happen here, now we use these words, right, is that our neuron here is polarized, the outside compared to the inside. The outside's positive, the inside's negative compared to each other. We can call that polarity also. This is not the polarity of like one molecule, like a water molecule, like we've also used the word polarity, but it's a polarity of a situation where you have a separation of positives and negatives here outside this cell compared to inside, okay? What this action potential is going to involve is a depolarization. That means something is going to cause this to flip and then get fixed. Repolarization, right? Go back to where it was. So the flipping of it, the depolarization, is what the action potential is all about. And then the repolarization reverses that to let this nerve fire again. So all this will become more clear as we break it down in more detail, right? But this thing that we call a nerve impulse is an action potential. It's propagated. Here's this word that's commonly used here, right? What that means is it's moved along, propagated along the axons of neurons, Well, it's also propagated from the dendrites to the cell body and then along the axon eventually, but here it says, along the axons. And it's the result of local currents. What's that mean? It means like not your whole body is having these currents, just local. And a very small part of this action potential impulse is being propagated in a very small area to the next very small area, very small area. That's what that one means. Okay? That cause each successive part, each successive little area of the axon, but also of the cell body and the dendrite, as we'll see, right, to reach the threshold potential. Here's a new word that's a key word, threshold, right? That's going to become more clear as we go. So I said a lot there. Now we're going to break all that down. Okay, so here is now with a little more detail that you need to be able to know, and especially in terms of this graph that's going to come up here in a minute, right? Here we have a number associated with resting potential, negative 70 millivolts. That's never going to be in red ink, but this graph that we're going to show in a minute, so I'm going to put it here now, right? Here's the membrane potential. This is my payback for me having to read some of your bad handwriting when you do tests, right? So this membrane potential, what's that mean? How different is one side of the membrane than the other? And this has traditionally been conceptualized in terms of the inside compared to the outside. So like we said, when this neuron is at rest, the inside is more negative than the outside. So if this is 0, we'll make the origin 0, right? That means the situation when this neuron is at rest is down here, negative. How much more negative is the inside compared to the outside? 70 millivolts. Therefore, the resting potential is negative 70 millivolts. That makes sense? That means the inside is negative 70 compared to the outside. If you put, like it says, like it shows here, if you put a little electrode inside and measured the outside, it would be 70 millivolts more negative inside than outside. Okay, I think we've said that enough, right? Okay, so now what's going to happen when this thing is no longer at rest? When this nerve cell gets the message that, hey, we need you to send an impulse, an action potential, what happens to the cell? What happens to the cell as shown in this graph? That's where we're headed. Okay, so what this says here is that in this nerve cell, there's a nerve cell again, there's the axon, right? There's the dendrites. In this nerve cell, that there is a gradient of these two positive things, and this is the gradient. Higher sodium concentration outside compared to inside, higher potassium inside compared to outside, right? Now there's sodiums in here, it's just a higher concentration outside, so I'll put 2 out here compared to the 1 in there. There's potassiums out here, but there's just more potassiums, higher concentration inside than outside, right? Remember, it's you need to say higher concentration, not more, like I did, because it's not necessarily more. I guarantee you there's probably more potassiums outside this cell in the universe than there are inside this little cell, but there's a higher concentration inside than outside. Okay. You also have some other negative kinds of ions that are going to play a little part that we'll talk about later, but the main players by far are sodiums and potassiums. And again, what this membrane potential being negative 70 means is that the positives outside simply outnumber the positives inside, concentration-wise. So now, here we go. We mentioned that there are sodium-potassium pumps in this story. Well, the sodium-potassium pumps are in the membrane, right? Traditionally here, we look at this story in terms of a section of the axon. That's why they talked about the axon having an action potential and propagated down the axon. But this little diagram could also be of any part of the cell body or any part of the dendrite, but it's always the axon, so that's how I'm going to keep it. But just so you know, that these events that I'm talking about are also happening across the membrane of the nerve cell body or across the membrane of the dendrite. Okay, so what's the situation here? Well, in this membrane, again, we have sodium-potassium pumps, but we also have channels of two different kinds. What's channel mean to you? Here we're reviewing again. It means a facilitated diffusion protein, right? A protein that helps facilitate diffusion. And we have them for both sodium and potassium. So in this diagram, they show the sodium-potassium pump. They really ought to show another purple protein that is a channel for both sodium and potassium. So what does this diagram show? I'll put it out here. If we have more sodiums outside than inside, and we have a channel that they can go through, they're going to diffuse into this axon or into this cell body. Right, I made it a cell body again. And if there's a channel for potassiums, and they have a higher concentration inside, they're going to diffuse out. What's the sodium-potassium pump going to do? It's going to reverse those, right? And pump the sodiums back out and pump the potassiums back in, therefore maintaining this gradient. Okay, so you say, well, that doesn't make sense. Why all the energy being spent to just keep something like it is? Because it's in the not keeping it like it is. What is like it is? Polarized. It's in the depolarization of this system that we have this thing called a nerve impulse. Okay, so now we'll expand on that. We'll have to go slowly here because this is tricky unless you break it down like this, I think. All right, so here's another aspect of these channels. I'm going to draw another one right here. They have a gate. Remember, these are proteins. These are globular proteins. They're very intricate in their shape. And some parts of that protein might be like a gate that can open and close. And therefore, this channel might not be and isn't open to sodiums all the time. The gate might be shut. And the same with this potassium channel. It's a gated channel. And that gate can open and close based on certain situations, and that's where we're getting to. Okay, so with that background, we're going to watch these in class. Okay, I'm just going to diagram